The broadcast is now starting. All attendees are in listen-only mode.
Okay, welcome everybody to what is the fourth day of our conference. Glad to see you all back. Let's have a look. Attendees are slowly arriving now. So after yesterday, we had such a great session despite some technical issues on the Vaughan Bryant session beyond miscellaneous. And so the organisers had a bit of a wind down at the end, and we've reorganised today's session to put Vera's talk in that hopefully she'll be able to make later. But before that, we've obviously got a great session coming up looking at Precambrian material. Before that, we've just finished the board's incoming meeting, the ASP board. Lot to chat about, lots of exciting things. We'll see if we can actually fulfill all that during our 10 years on the board. You'll see the slight changes on the new handout that's uploaded. But other than that, then there's so the revised schedule is on the new handout. Again, I'd like to promote the fact that there will be awards at the end on Friday. So all students who've presented either a poster and lightning talk or a standard talk will all be in contention for a prize of 2 years annual membership to the AASP Palynological Society and also $250. So nothing to sniff at there. Excellent. I think I don't think there's much more for me to go on about so much. There will be a third student mixer after we included another one after the awards, which we will send out a link to towards the end. We're just waiting on deciding how long the awards session is going to last, because we have a few awards and some exciting awards to give, other than just the great student talk and poster awards. So without further ado, I will hand over now to the session chairs, Evelyn Sanchez and Thomas Fairchild, who have come up with this next session on Precambrian palynology. And before I say any more about it, I'll let them take over and introduce the session. So over to you.
Well, thank you, Stephen, and hello, everybody. I hope you all are healthy and well, you and your family. Welcome to the Precambrian palynology session. We are very happy that you came. We are going to start our meeting with the lecture review of Australian Precambrian palynology by Dr. Kathleen Gray, and then we move on with another 5 standard talks. Unfortunately, Dr. Jaeger will not join us today, so the last talk will not occur. For the presenters, please keep your presentation ready to go so we can keep on the correct time, okay? Please, thank you. And now I'm very pleased to present to Dr. Kathleen Gray, although I think she doesn't need a presentation because she's an international Neoproterozoic star. But I'm very pleased to present to Dr. Kathleen Gray from the Geological Survey of Western Australia. Certainly, when we talk about Neoproterozoic microfossils, biostratigraphy, her name shows up once She was a pioneer in proposing an acritarch-based biostratigraphical scheme for neoproterozoic, and her proposal is nowadays worldwide accepted and worldwide applied. So certainly she's an authority regarding neoproterozoic palynology. She also has experience in older sections throughout the Precambrian record, including Archean palynology. Very nice, very interesting. So thank you, Dr. Kathleen Gray, for accepting our invitation. We are very happy to listen to you now. So please feel free to share your screen and start your lecture. Thank you, everybody.
I don't know if this is, oh, okay, that's good. Can you hear me?
Yeah.
Right, okay. I'm not very good with all this technology, so I don't want to stuff up too much. Do I grab my file now? Is that the story?
Yes, that's right. If you could bring up your presentation, that would be perfect.
Is that under sharing?
It should just be, we already see your screen, so if you just bring up the actual PowerPoint presentation, we should be able to see it.
How do I bring it up though?
Is it opened on your screen?
Yes.
Okay, if you put it into presentation mode, so if you make it the full screen.
Yep. How's that?
Yeah.
Yeah, so we see it, but we see your presentation view. So at the top, if you click display settings.
Right, well.
Do you see the, there's a little bar that says at the top, it says display settings.
It says what?
Display settings. Yep, just to the, at the top.
Sharing, webcam, audio, attendees, handout I've got.
Just at the top of your actual presentation, it says display settings. Yep, the next one over.
Where does it show it?
Yep, there. Yeah, right there where your mouse is. If you click on
display settings.
Yes, yeah, if you click on that. And then if you hit swap presenter view and slide.
Yeah. Is that what I'm supposed to go for?
Click that one.
How's that?
Yep, that's perfect. Thank you.
Right. I'm not quite sure what I'm doing here, but anyway, I'm sure we'll get there. Have you got all the other screens showing over the slide view or not?
No, we just see the title slide. Yep, okay. That looks perfect. Go ahead.
All right. Well, considering the subtitle, this probably ought to be 3 billion years in 30 minutes. You'll appreciate I can only really do some highlights, and I really will have to get going very quickly, or I'll be lucky to get through it. So can you now see the preparation flowchart and protocols slide 2?
Yes, we see it.
Yes, and I thought I'd just put these up as a warning for anybody who's thinking of working on Precambrian. One of the problems is that the rather vigorous methods that you tend to use on the Phanerozoic stuff doesn't work with some of the Precambrian stuff. They're too fragile. So there's this very complicated process where you have to go back to the very old techniques we use and keep using gravity settling and so on, and that way you do get good results. Yeah. If anybody wants to know about it, I can send you, or I can link you to a copy of my report on how to do it. And I've certainly had excellent results, and I've compared them with before and after results in the past. So it's well worth knowing about it. Another thing you need to be aware of when you're working on Precambrian is taphonomy. And taphonomy if you don't know much about it, it's the degradational history of preservation of preserved material. It's what happens during death, decomposition, burial, and it controls things like folding, compression, and diagenesis, which can change the colour. And of course, the colour is quite significant. It does indicate the degree of thermal maturation. The TAI, a useful index tool for hydrocarbon exploration. So these are things to notice. And you have to consider taphonomy as well when you're identifying specimens. One of the problems is that you get something like this, where I've got the top one is a very nicely preserved specimen, and then the one further down is a not so well preserved one. It's obviously undergone a higher degree of alteration, but it is the same species. Both of them are Cynarium conoideum, and you need to be conscious of that sort of connection as you go through different degrees of taphonomy. I've come across quite a few things where the same species has been assigned to several different species because the taphonomy is not being considered. Another thing that's very significant in any Precambrian work is the criteria for identifying ancient life. And this goes to what's known as ECRE and is attributed to Carl Sagan. Extraordinary claims require extraordinary evidence. There's a lot being written about it, particularly in Schopf and Klein's Proterozoic Biosphere. And there's also a paper in Precambrian Research in 2007 that Ken Sugitani wrote, and I was an author on, where we paid a lot of attention to establishing our claims for some of the Archean fossils I'm going to show you. And the basics are fossils must be in rocks of known provenance, indigenous to the rock. They must be formed at the same time as the rock, and you need to look at the criteria for how you're going to establish these things. They must be formed in an environment that's suitable for life. It's no good giving me a chunk of granite and trying to convince me that I'll find fossils in it, because it's most unlikely that anything would be living in the molten lava. And it must also be of an assured biological origin. That's a very difficult criterion to prove. But you need to be careful if you're making morphological comparisons, because you get things like these biomorphs, which you can see down here, which are actually crystals that look like they're biological, except these actually are a lot more cup-like things that sit inside each other. But you can see, if you're not careful, they could be mistaken for this, which is a living cyanophyte from one of the nearby lakes. And of course, one of the classic examples of this was the Allen Hills so-called bacteria that came from a Martian meteorite. And there are all sorts of clues there that that wasn't really biological. One thing being the size, it's only in nanometers instead of micrometers. And you have to look at it very carefully, and you have to know what living equivalents look like in a way in order to interpret structures like this and decide whether they're fossil or not. The other thing, which is a big surprise to people who've worked on a lot of Phanerozoic stuff, is the amount of time you're dealing with, an enormous amount of time. And it often fools people because so much can happen in the sedimentary environment in the period, in the subdivisions of time. This is the International Subcommission's division of the Archean, and I'll be working my way quickly up through from the Archean to the Cambrian boundary. And I'm trying to pick out what methods we use. So these symbols, the little brown ones mean that the work was from thin sections, and the green stars means that palynology was carried out. It's not always easy to carry out palynology on the material. You need the amount of material. In the first place, it needs to be suitable for palynology preparation. But I think you can see from this diagram, some of the older stuff, there's no palynology at all. The one I'm pointing out here is one I will be talking about, which is the work I did with Kenshiro Sugitani. And he's submitted a poster, which is probably the best way of finding out more about this. And it's not really till you get up to the Proterozoic that palynology comes into its own, but there are one or two key examples of using palynology in a slightly novel way that I'm going to talk to you about. Now, for those of you unfamiliar with Western Australia, this is the Pilbara region on the sort of northwest coast of Australia. If you're a Mesozoic palynologist, you've probably heard of the Pilbara Shelf, which is up here, which is the Northwest Shelf, which is where a lot of the Phanerozoic petroleum and gas comes from. But being a Precambrian paleontologist, I'm more interested in the Pilbara region, which consists of a lot of narrow greenstone belts that have got trapped between granite plutons, and that's where we find the microfossils. And some of them, there are several different groups that I'll talk about in a minute, but the ones I'm particularly interested in are these ones in the Strelly Pool Formation, and these others in the Farrell Quartzite. You'll see they're different ages, and I'm still worrying a bit about whether some of the stuff mapped as Farrell Quartzite is really Strelly Pool Formation, but I'm not being able to persuade anybody to go out and redo the mapping. However, you'll see why there's a bit of concern about it when I move on. So in the Paleoarchean, there are quite a few examples of things claimed as microfossils. A lot of them, except for this Strelly Pool Formation stuff, are spherical or tubular microstructures. Many of them are of ambiguous origin, and many need further studies to confirm their biogenicity. Probably the oldest of these things in the Dresser Formation, filamentous and spherical, that were described by Eugenio, and they really need further work. Another important one was in the Mount Ada basalt, described by Aramic et al. Received a lot of bad press because the locality wasn't exactly known. We think we subsequently found it again, but we haven't been able to find the right horizon to reproduce the results. But looking at this, to me, I'd be fairly convinced that that is biogenic in origin. It's more than can be said possibly for the ones in the apex basalt, which were highly criticised by Brazier. This was Bill Shopp's material, and you may remember there were huge debates about it for a long time a few years back. I have seen the original material in thin section, and I do think a lot of it's biogenic in origin, but it's very badly degraded, and it still needs more work to be sure. There are other units in the Paleoarchean, the Panorama Formation, the Eurobasalt, and the Kangaroo Caves, which also have claims to have biogenic structures in them. Some of them are not that convincing, but they may well be. The one I'm most interested in is the Strelly Pool Formation, which is around 3.4 and a half billion years old. These are the specimens that were found by Ken Sugitani, and the Strelly Pool Formation is really a very, very interesting unit because it's got spectacular stromatolites in it, beautiful conical ones, these little egg carton ones, and much larger ones, which are up to 1 meter in diameter. And in fact, 1 meter in height, sorry. And in fact, some of them have the axial zone, which is characteristic of conical stromatolites and can be seen in modern stromatolites, or the little pinnacle mats that you find in hot springs and so on. And I'm fairly convinced that these stromatolites are biogenic. So it's interesting that these microfossils occur along with the stromatolites in the same unit. And hopefully this will give you an idea of what the microfossils look like. They're consist of a sort of central body, and they're slightly elongate in shape. And I'll show you some more details, and you can also look at Ken's poster. So that's the Dixon Island stuff, and so on, which is Mesoarchean. And when we reach the Neoarchean, you'd expect there to be a lot more in the way of microfossils, but in fact, we don't see that many. This is probably one of the better ones. It's not that good, but it's a septate filament in the Fortescue group. I haven't really seen many others, except when I looked at this stromatolite. Again, there's loads of stromatolites in the Tembiana formation. These are little finger-sized columns, and when you look at thin sections of those, they've got a lot of tubular structures in them, and a few spheres and a few clustered spheres. But you do have to be very careful, because this is also present, and it's ambient pyrite, where a pyrite grain's moved through the colloidal chert and left a tubular opening behind it, easily mistaken for microfossils, but another warning of being careful working on the stuff. However, the Strolly Pool formation has much in common with the Farrell quartzite, which is a much younger unit and is about, well, we know it's younger than 3 billion on the current mapping, but as I mentioned earlier, I wouldn't be surprised if it turned out to be Strolly Pool chert. But for the moment, this is really the most spectacular Archean material that I've seen. I was really surprised when Ken brought it in to show me. And what we found in thin sections were tissues. And this is a reverse of taphonomy, because the taphonomy is showing you tears in the tissue and fold structures and embedded spheres. If you saw this in the Neoproterozoic, you'd say, oh, it's bacterial mat that's folded up. You also see spheres that are broken with folds in it. And we saw clusters of small spheres and much larger spheres, again showing taphonomic alteration to those. And I don't think you'd see the effects of taphonomy in crystal structures. And you also see that they're hollow spheres. They look like some of the ones you'd see in the Neoproterozoic in similar circumstances. And then we've got what we call the lenticular ones, which are very similar to those elongate things in the Strolly Pool formation, often with this dark shadow across them. And this was a nice photo reconstruction that Ken put together, and you could actually see that this flange-shaped structure sometimes has reticulation on it, and here it seems to have striae. One of the most amazing things is that we see colonial structures all linked together, and Ken's got some good illustrations of those in the poster. When we published the paper, we were highly criticised and informed that these couldn't be integral wall structures. They were just fine-grained particles that have been pushed apart, pushed away from the areas they're in by crystal growth. And after thinking about it for a while, I thought, well, what happens if we treat it palynologically? And of course, the answer would be that all you get is a pile of disarticulated particles. So we did try palynology on it, and we got anything but disarticulated particles. We still got the large tissues with folds, structures. We got small spheres. We got the large spheres, integral things. We also got, not very well preserved, I will admit, but we also got lenticular structures from the palynological preparations. And I know Ken has got better examples than the ones I have photos of here. This also allowed us to do a lot of high resolution microscope work in order to do a three-dimensional reconstruction. And when you view them in mid-focus, you can see that the central body is hollow and that the flange-like structure's tapering. And then you can play by moving up and down the focus on the microscope to get that sort of shade of the flange appearing. And in a few rare instances, we also got polar views. So you can see the flange isn't necessarily just circular around them. Now, to me, this is a very highly organized level, a very high level of organization. And it goes back to one of the talks the first night of the meeting when somebody said, are there green algae in the Pilbara, in the Archean? And I think the Pilbara might well be a candidate for that. I'm sorry I can't spend a lot of time on these. They're very interesting, but I have to move on to the sedimentary basins. Australia has a wealth of Proterozoic sedimentary basins. I've visited most of them, collected from most of them. And so what I'm going to do is, first of all, I'm going to mention the Irahidi Basin and this area in here, which is known as the Ashburton Fold Belt and has the Turi Creek and Wailu groups in it. This has a formation called the Frere Formation, and you'll see why I'm comparing those. I'm then going to talk about the MacArthur Basin, which is this area here. I've done quite a bit of work on that recently, and I'll carry on from there to the Victoria and Birindudu basins, and also look at a unit that we found in Western Australia that's a rather disconnected unit called the Badgerada Group, which sits between the Yilgarn granite craton and the Darling Fault. And we still don't know a lot about it, but it does have some nice palynomorphs in it. Moving on, what's happened here? We should get time, I hope, to talk about the Neoproterozoic, which is the area outlined in red and which was the subject of my thesis, and which I think shows what can be achieved with palynology and biostratigraphy in the Proterozoic. This is the general division of the Paleoproterozoic to the mid-Paleoproterozoic. Here's the Turi Creek group, the Duck Creek Dolomite, which is part of the Wailu group, and the Frere Formation. And then I'll be going on to talk about some of these units here in the MacArthur Basin and associated with it. And you can see a lot of this work is based on thin sections with just a little bit of palynology. So here are thin sections made of these three units. This is a fairly recent publication by Barlow and Van Kranendonk on what is supposedly the Turi Creek group. And you can see it has one or two nice microfossils in it, which can be identified as Kakabechia. The Duck Creek Dolomite is considerably younger and also contains the same sort of thing. And it has Huroniospora and Gunflintia and Eoastrian, which is this type of thing as well. And so does the Frere formation in the Irahidi group, which was described by Walter et al. In 1976. Again, it has Eoastrian and Kakabekkia in it. And this raises the issue of, are these units containing similar microfossils because they're similar fascies? I should also point out that these fossils were described from the Gunflint Chert in Canada. This still remains quite a mystery to me. I've not had much to do with it, but it certainly needs more work. Now we're moving on to the MacArthur Basin, and this is this large unit in the Northern Territory, and adjacent to it is a buried basin, sub-basin called the Beetaloo Sub-Basin, which I'll be talking about. Sorry, I've lost that. Ah, here we are. So this is what I'm going to focus on now. In the 1970s, the main focus in this area was on significant mineralization in the MacArthur Basin, in the MacArthur group. And there were a lot of mineral deposits up here. Some are now mines. And since then, interest has shifted to the Beetaloo Basin, where the younger Roper group has hydrocarbons, actual oil at about 1.4 billion years, which is quite spectacular, really. So this is now very quickly the stratigraphic chart, and if anybody wants copies of this and so on, I can send them to you later. But again, you can see there's not that many units that have had palynology work on, a little bit. The pioneer in this area was Marjorie Muir and some of her research students, and they looked at some of these units in the MacArthur group and illustrated quite a few fossils, mainly in thin section. Later on, when live oil was discovered in Europinga 4, which is this drill hole here, and Jamison 1, several oil companies investigated the hydrocarbon potential of the mostly unexposed Roper group, and that overlies the MacArthur group. I was asked by any one of the oil exploration companies who had an interest in the area, would I investigate the biostratigraphic potential of several selected drill holes from the upper Roper group. And I made what can only really count as a preliminary study based on 52 samples from 7 of the drill holes. And that worked out at about 2 samples per formation per drill hole. So there's still an awful lot more work that could be done. However, I feel the results were very encouraging and actually built on some of the earlier work, such as this, which was one of Marjorie Muir's students, Pete, who in 1978, this was from his thesis work, illustrated a very large number of specimens from the Sherwin Creek iron ore deposits. The unit that he referred to as the MacMinn formation is now the Sherwin and Kyalla formations. And he looked at 367 samples. However, nobody seems to know exactly where they were. A few of them were palynological preparations, and he reported this diverse, abundant assemblage. But however, only a brief paper was published, and the details are still in his thesis and difficult to get hold of. However, these very promising results, for some reason, just weren't followed up. And I think it's a great pity, because I think it would have helped greatly in correlating all the various units in the MacArthur Basin and the Beetaloo. This is the stratigraphy of the Roper group, which is the more interesting of the two as far as I'm concerned. The Kyalla and Valkyrie formations are active targets for hydrocarbon exploration. And when I say they're active, they've got live oil in it, and you can actually see the drill core and run your finger over it and get oil on your finger out of the drill core. There's been a bit of exploration work, but it's never really reached production level. But I also think there's a lot of work still to be done on correlating them. One of the earlier studies was carried out by Javot et al. And Javot and Noel. They actually looked at MacArthur Basin drill holes, but in the Roper group mainly, the lower part of it, the Mainaroo, the Crawford, the Jalboy and the Corcoran. And it was mainly a paleobiological study. They recognised the large process-bearing acritate with these rod-like structures and little processes on the end, to Paleoplana, which is really quite a well-known form. And they recorded about 34 taxa, some of them new, and together with other evidence, other acritates, they thought this was possibly the evidence for the earliest eukaryotes. However, I think this can probably now be pushed back a bit further, but the work still needs to be done. And they also thought that the control was mainly environmental. They never really published the biostratigraphic distributions. They didn't really do a range chart as most biostratigraphers would know it. And it was very difficult to extract the locations for some of their material. Their interpretation implied that the palomorphs were unsuitable for biostratigraphic correlation. So again, the results were never really followed up at the time. And it's a pity because a lot more could have been done with it. The Valkyrie, well, this is the work that I did for ENI. And
5 minutes, okay?
Sorry?
You have more 5 minutes, okay?
Oh gosh, I'm nowhere near through. This is the Valkyrie formation, really nicely preserved material. If anybody knows what this is, there's some very unusual looking things I'd be interested to know. And the Kyalla formation, again, with some really nice looking, particularly aggregates. And the uppermost of the units I looked at, the Chambers River formation, you can see the preservation's beautiful and some really exciting material in there. And I was able to put together a preliminary range chart. And I think this is something that could be followed up. Roper group palomorphs are abundant, well preserved, and include undescribed taxa. They've good potential for correlation, but we need a lot more work on them. And they could provide a basis for developing a correlation scheme that would include not only the hydrocarbon-prone Beetaloo subbasin itself and correlations between the drill holes, but it could be correlated to the MacArthur Basin and extended to other sedimentary basins. I was given some slides from another set of units called the Victoria and Birrindudu basins, the Angularri siltstone, the Roper group and the Kunja siltstone. And a student called Tara Lewis did an honours degree on this. Here you can see the general stratigraphy. I'm going to have to go very quickly, I'm afraid. And here are the basins in question. And here is the assemblage. The Kunja siltstone's got these enormous filaments that are about 100 microns in diameter. Many of these others look familiar from what I've just shown you in the Roper group. So there's a good potential for correlation there. And again, that little unit I mentioned trapped between the Darling Fault and the Yilgarn. Another student from Curtin University, Rhys Nichols, did his honours thesis on it, and we got really beautiful specimens out of it. Again, very similar to the ones we're seeing typically in some of this Mesoproterozoic, and I think capable of more systematics and correlations. That's where the Badgerada group is, so it's quite a long way from the other basins. I've got to go very quickly on to the Neoproterozoic, and this gives you an example of all the areas I've worked on and visited in about the last 50 years. A lot of drill holes. I think altogether I must have looked at, I've forgotten how many now, there'll certainly be over 100 drill holes. And I've processed and looked at over 2,000 samples. I did a lot of the processing myself for my PhD work. So you can see this is the area that was covered by Neoproterozoic sediments, the Adelaide Rift Complex and the Centralian Suprabasin. And this is published in Gray 2005 in considerable detail. And I've also looked at the units further down. This is the PhD work was mainly on the Pertutatica and corresponding units in about 5 tectonic units, and was Ediacaran with a little bit of Cryogenian, but I've also worked on Latonian and Cryogenian. So this was the thing that used to be called the Bitter Springs Chert, and due to stratigraphic revisions is now the Johnny's Creek Formation. And another new unit, the Wallara Formation, which has Cerebrosphaera buicki, which I'll mention in a minute. Then we get the Sturt, the lower of the two glacials. We have a interglacial period called the Arauca Formation. There's other formations in the other basins, and the Elatina glaciation of the Cap Carbonate. And then the Pertutatica Formation, which was I was able to zone quite successfully for my work. So I'll have to dash on. This is what the Bitter Springs Chert or Johnny's Creek assemblage looks like. Lots of Mixococcoides, various spherical things and septate spheres. This can also be obtained by palynological extraction. Higher up, the Wallara Formation and all its equivalent members have this assemblage, mostly lyospheres and so on. I like to call it the Cerebrosphaera buicki and assemblage, and it more or less equivalent to the first appearance of a stromatolite by Callia burra. This is by Cerebrosphaera buicki, that's the type, and the SEM thing, which was illustrated by the author, Butterfield, from Spitsbergen. These are from the Officer Basin and the Amadeus Basin and the Adelaide Geosyncline. They nearly always appear at the same horizon in each of these drill holes. And it also occurs in the Grand Canyon in the Galaros Formation, and was illustrated in Naju's thesis. So in my opinion, there was a paper by Porter and Reedman on one lot of Officer Basin drill holes, and they suggested that Cerebrosphaera buicki should be placed in synonymy with Cerebrosphaera globosa, following Sergeyev and Schopf. But I must admit that that's something that needs work on, and I'm not at all convinced. I think some measurements need to be done to verify that. It really is an important first appearance because it's a potential basal marker for the Cryogenian period, coupled with the first appearance of Callia burra, and there's also an isotope excursion that fits with that. Then we get the interglacial assemblage, which, looking at the lithology, I would have thought was going to be spectacular. I collected loads of samples, nearly all of which were barren, until I got just nearly to the top, when I discovered a couple of samples that had this thing in it, hundreds of them, and it's some sort of Vandalosphaeridium, and I think it's a new species. It's not published properly, and it really needs proper systematics doing on it. It's then followed by a liosphere interval, which I'm not going to show in detail because it's the same old liospheres associated with Optoidrixium truncatum. And then you get the ediacaran acanthamorph assemblage, which is very different. And you can see here all the spiny acritarchs. I was able to put together a range chart, which has turned out to more or less stand the test of time. Sebastian Willman and Margosha Mokshadlauska did some more work on some of the drill holes from the Officer Basin. And although there was some tweaking of systematics and so on, and some of that still has to be collated in with the broad data, most of it still seemed to work quite well. So we had 4 zones of these acanthamorph acritarchs, and they don't really appear until just after the Ackermann impact ejector layer, which was a big bolide impact, which threw out debris that you can actually see in the drill holes. Before that, you've got liospheres, and they decline as a percentage once you hit the Ackermann impact layer. But fairly recently, I spotted this, which is one spiny acritarch, one species of spiny acritarch, and it's the only one that I've found below. So it's a bit like thinking of the mammals being around but in small numbers before the Chicxulub impact, and then they took off and were able to diversify, and we're seeing the same sort of diversification occurring in the spiny acritarchs. This is the Appendisphaera minuta, the type specimen in the Amadeus Basin, and these are the new ones in the Officer Basin. There's about 20 in a single slide, so they're not common. They're very delicate, so I'm surprised that some of the more robust ones aren't found in these slides if they are actually around at that period in time. Again, it's something that needs further investigation. So this is the pictorial thing. Here we have the Johnny's Creek assemblage, the Cerebrosphaera buicki, the glaciations, the Arauca with the Vandalo at the top. We have the Elatina glaciation, mainly followed immediately by liospheres, with perhaps the 1 or 2 of the acanthamorphs, and then we go up into this huge assemblage of acanthamorphs that can be divided into 4 separate zones. For the benefit of people who are interested in reconstructions for the basin, I was able to draw this up showing how these drill holes correlate, and there's lots more drill holes that haven't really been investigated that need to have palynology done on them and see how they fit together, particularly in the South Australian Officer Basin. So time for conclusions. Sorry, I've probably overrun a bit. This is really just a sort of quick sweep through everything, and I hope I've convinced you that Australia has an amazing Precambrian palynological record, as well as the microfossils in chert. There are tantalizing fossils in both the Archean and Proterozoic that require paleobiological study. For example, what are they? Are some of them green algae? What else could they be? There's much work still required on systematics and data collation. The potential for the development of biostratigraphic correlation is a useful tool in the Mesoproterozoic and Neoproterozoic is particularly good, and I think needs further investigation. And as can be demonstrated in the Ediacaran, correlations work well when combined with lithostratigraphy, isochemostratigraphy, and stromatolite biostratigraphy. We need about a 3-day symposium to go through all the evidence, so you'll just have to accept my word for it that it's working quite well, but our mappers at the Geological Survey would corroborate my statement. And there's much scope for using palynology as a correlation tool in the Precambrian. and in Precambrian rocks for both mineral and hydrocarbon exploration. And there are still kilometres of drill core stored in drill core libraries available to sample. So I want to say thanks to a cast of several hundred paleontologists and geologists who've worked with me over the 50 years of my career and helped me to put my story together. There are far too many names to mention. And I hope this very rushed review will encourage some of you students and early palynologists to consider the possibilities of working on the Australian Precambrian. There's heaps of work to do. Unfortunately, I know some of you know that I've been battling cancer myself for 3 years. I'm doing quite well at the moment, but I'm not going to be around that much longer. And I would really like to pass the baton to other enthusiasts, having heard the stories about Vaughan and how he teed people up to follow on with his work. So thank you very much. And if anybody wants more information, please contact me and I can put you in touch with the literature and with this presentation and anything else you would like to use. Thanks very much.
Thank you very much, Kathleen. Amazing talk, amazing lecture. I'm sorry to interrupt you. We are a little bit late, so no time for questions, but I think if you have any questions, you may send by email to Kathleen Ray, right, Kathleen? Okay, thank you very much.
Not next week, because I've got my round of medical checks next week, but the week after, I'll be happy to start passing on information if people need it.
Okay.
Wow.
All right, okay. Thank you very much. And now we move on to the standard talks, beginning by the presentation by Ismael Acosta and Claudio Gaucher. The presentation is Organic Walled Microfossils from the Passo Severino Formation, Paleoproterozoic of Uruguay. So, Ismael, please.
Can everybody hear me?
Yes.
Is my screen shared?
Yes.
Okay. So, okay. Hello, everyone. I will stay with my camera off to improve my connection. I hope that this is fine. Good morning, good afternoon, or good night, wherever you are. My name is Ismael Acosta, and I am currently an undergraduate student doing a bachelor in biological sciences at the Facultad de Ciencias, Universidad de la República in Montevideo, Uruguay. I want to thank the organizing committee for allowing me to present this work, titled Organic Wall Microfossils from the Passo Severino Formation, Paleoproterozoic of Uruguay. So, to begin with, the characterization of Precambrian life is difficult and restricted, and Uruguay is no exception. The oldest evidence of life in Uruguayan territory dates back to the Archean and consists of stromatolitic remains. As we move further back in the geological record, the distinction between fossils and pseudofossils becomes increasingly difficult. In paleontology, especially in Precambrian paleontology, the concept of biogenicity is extremely important, and last talk was especially very good about that. It can be defined as the attribution of biological characters and origins to structures of the natural world. Biogenicity is particularly complex in Precambrian rocks due to various metamorphic processes that can alter the fossils, and that makes the interpretability and testability of biogenicity quite challenging. Therefore, we need the evidence of Precambrian life to be positive. That is, it must affirm the biological origin of the objects. Biogenicity cannot be determined by neutral evidence. That is, the sample may or may not have a biological origin. This kind of statements should be considered inadequate and require further analysis. Moreover, interpretations of biogenicity by negative reasoning or inferences by default, that is, claiming biological nature solely on the basis of discarded mineral origin can lead and has led to significant errors. So, the Paso Severino Formation consists of a series of volcanosedimentary successions metamorphosed in green-schist facies. In particular, a metasedimentary sequence composed of carbonaceous black shales and dolostones are highly concentrated in organic material. These rocks were metamorphosed during the Trans-Amazonian orogeny. Several papers have determined the age of this formation. Interweaved acid volcanic rocks yielded a UPV shrimp zircon age of 2,146 million years, accepted as the depositional age of the unit. Moreover, carbon and strontium isotope measurements for carbonates are consistent with an age of 2150 and 2100 million years. Apart from that, carbon isotope values reach plus 11.6 per mil BPDB, typical of the Loma Gundi carbon isotope excursion that occurred between 2220 and 2060 million years. The objective of our work is to study and identify microfossils in the metasedimentary rocks of the Paso Severino Formation by applying different micropaleontological techniques. In particular, paleontological macerations, thin sections, combined trans and API illumination microscopy, and scanning electron microscopy. Samples were extracted at the localities shown. Specifically, we selected 3 main regions for the formation with different lithologies. One more associated with shales, another exclusively carbonates, and another with a mixture of shales with dolomites occur. In total, 19 samples were obtained, from which 19 paleontological macerations, 3 thin sections, and 35 microscope slides were made. One of the groups observed were spheromorphs, highly carbonized, some with folds and even mineral incrustations. Spheromorphs are highly variable in size, between 10 and 70 microns, some even with internal subcellular divisions, like this one over here. Within the group of spheromorphs, it was possible to taxonomically identify several Leiosphaeridium specimens, mainly by scanning electron microscopy, as shown in this presentation. This taxon is characterized by this relatively simple and smooth organic wall, with a spherical morphology without ostentatious cellular processes, with an approximate size of 50 microns. A significant number of polygonal acritards were found, particularly those with pyramid and rhomboid shapes, many of them similar to the Proterozoic species Octoedrixium truncatum. Note that the walls are indeed organic, but are highly carbonized to distinguish any internal cellular features. On the other hand, some specimens were catalogued within the group of acanthomorphic acritards, with microfossils composed of several subcellular processes of different sizes, which would imply a set of microfossils with a certain degree of cellular complexity. However, many of these structures could have formed during diagenesis, so it is necessary to take precautions when classifying these structures taxonomically. At a more detailed level, these acanthomorphic morphologies are relatively folded, and it is complex to assign them a taxonomic category. However, the ubiquity of these morphologies and in the lithologies studied is striking and probably requires further analysis. Moreover, specimens of colonial spheroids were also found, as can be seen in these photographs. Especially, this group of spheroids is formed by 7 spheroids arranged in 2 rows of 3 and 4 spheroids. At the level of scanning microscopy, it was not possible to find relatively well-preserved possible colonial spheroids. One of the most striking morphologies was the high occurrence of branching structures, mainly those with 3 branches. These branching structures are difficult to catalog, but some of them resemble in size and morphological arrangement of the Proterozoic genus Eoastrion sp. Other striking morphologies are these structures similar to the genus Cacavecchia, with their characteristic umbrella-like structures and terminal bulb. Both these Cacavecchia-like structures and the previously shown Eoastrion-like structures would indicate a possible affinity to bacterial groups. It has been hypothesized that the different sizes and shapes of Cacavecchia could be associated with different ontogenetic stages, although, once again, diagenesis could have produced these morphologies. On the other hand, very few filament structures have been found. Those structures that could indicate the presence of filaments are poorly preserved to be assigned a taxonomic category. Here's a sequence of photographs obtained with optical microscopy with API illumination to show that these filament-like structures are indeed organic matter, but they are poorly preserved to be interpreted taxonomically. These filamentous specimens are the only ones found, so they constitute a rather low sample to be able to determine if they belong to any known group. In conclusion, we have discovered a relatively diverse paleomicroflora similar to other Paleoproterozoic assemblages, such as those found at Gamflin and Franceville, although Paso Severino's microfossils are much more carbonized and consequently less preserved. To our knowledge, these microfossils represent the oldest body fossils so far described for South America. Thank you very much for your attention, and I thank again the organizing committee, the Facultad de Ciencias, and my supervisor, Claudio Gaucher. I will be happy to answer your questions now.
Thank you, Ismael. We are still a little bit late, so no time for questions, but the email of Ismael is there in the screen, so please, if you have any questions, send to him. Okay, let's move to the next presentation. And now we are going to listen to Paul Strother, a possible holozoan palynomorphs for the 174 million years non-Sook shale. Paul? Paul, can you listen to me?
I think we're just waiting to connect to Paul's screen. There we go.
Hello, Paul, please. No. No, we cannot listen to you.
You're unmuted at our end, Paul.
Now, no?
Oh.
Oh, I think we might have just heard you very quietly, Paul. We don't hear you now.
All right, how about now?
Yes,
With
we can
audio.
hear you now.
You can hear me now?
Yeah, you're a little bit quiet, but we can hear you.
What if I go closer to the machine?
Yes, that's great. We can hear
Is
you
that
much
okay?
better now. Yeah, did you see the request to share your screen?
No, where does that request come from?
Oh, it should have popped up as a little box. Sometimes it gets stuck behind other things, but I can send it again.
I think I'm sharing my screen. Let's see. I'll try turning on the actual presentation, right? Okay, here, show my screen. And I have to go to slideshow. How's that?
We're just seeing your webcam. We're not actually seeing your screen.
Okay, I thought I'd show main screen. How's that?
There we go. Yeah.
Is that working
Looks
for you?
perfect. Yep, that looks perfect. Thanks, Paul.
All right, sorry, it's such a mess, but, and I've now got my, hold on a second. No, that didn't work. How's this? Is this still okay?
Yes, that looks great. Go ahead, please.
Okay, so talking about some kind of overarching problems in Precambrian palynology, kind of what we would like to do. Oh, that's not right. That is, we would like to be able to define good taxa. We need to, our biggest problem is the determination of the systematic affinity as the biological association of various taxa that we have. And it looks like my machine is on some kind of auto screen where it's automatically changing from one slide to the next. So I don't know why that is a problem with the display. And then also
Paul,
one of our goals.
We actually lost your presentation, Paul.
You lost.
We just see you again. Yeah.
Main screen.
There we go. We've got you back.
All right. Okay, sorry. Anyway, ultimately, we would like to have some kind of sense of reconstruction of both spatial and trophic ecology, that is the distribution of organisms and what their ecological role might be. And I've written down resisting the temptations of uniformitarianism. I probably won't have time to go into this too much, but basically the point is that not everything is just green algae. And I'm working in a different kind of deposit. These are non-marine deposits that are about 1 billion years in age. So we're not looking at the kind of the stuff that Kath Gray just went through, which are based primarily on looking at marine sequences. This has been an outline of the talk, although I'm going to have to cut it. I'll have to talk fast. We're all way over time. A little bit about the biology and geology of the Nonsuch Shale. That's the deposit that I'm talking about today in terms of these interesting clusters, and focusing ultimately on a particular type of cluster, which we refer informally as the morula form. And we want to compare this to the recently described multicellular protist Bicellum brazeri that we published just in June, and compare this to the life cycle of something called Triolimex fragrantissima. It's a holozoan protist that is in the lineage leading up to the animals ultimately. And these are interesting organisms studied by modern molecular biologists and phylogeneticists because they contain the genetic or the genomic precursors to complex multicellularity that occurs in animals. This is a diagram from the work of Sarah Slotnick et al., who's now a professor at Dartmouth University, looking at some of the cores that are drilled through the Nonsuch Formation, but going through the sequence of the Oronto group, which is a volcanic sequence, but is topped by a sedimentary cover that includes conglomerates and ultimately a thin layer of a lake deposit called the Nonsuch Formation, which is a dark gray shale and siltstone, and that's topped by the Freda sandstone, which is a very coarse and very thick, several kilometers thick red oxidized sandstone. And this all occurs on the south shore of Lake Superior in the mid part of the United States, mid-continent, well, between southern Ontario and the U.S., and the cores come from Wisconsin and the UP, or the Upper Peninsula of Michigan. They're associated with copper mineralization. Here's some examples of the sedimentary features that indicate that we're in very shallow water, including mud cracks and raindrop impressions, as we see in the upper right there, and sort of mat-like fabrics associated with the fine, thin laminae. There's some not a lot of myths there, but there are some sedimentary structures that could be interpreted. Others have interpreted as belonging to perhaps eukaryotic types of microbial mats. And there's some of our colleagues, Willie Taylor and Bas van der Schutsbrugge, scratching his head in perplexity looking at these shallow water indicators. This didn't come out in terms of the scale. This sequence is terrestrial, or this lacustrine sequence, and that's been known since 2000. I'm sorry, what is this? 1911 or so. Yes, Van Huysen and Leith. Anyway, there are some papers that have come out recently that have claimed that these are perhaps linked to marine sequences. Here's a reconstruction of the 1,050 million years old, and the Paleo Lake Nonsuch is about 800 kilometers away from the nearest ocean. So it's really hard to envision some kind of marine incursion going into this. So if there are saline conditions that are associated with the Nonsuch sequences, probably due just to increased salinity from evaporation or some such. These are some slides from a paper that we published in the JOLSOC last year, just to show you the kinds of things that occur in the Nonsuch assemblage. This is sort of an ongoing project in terms of getting the taxonomy done. They have the classic series of the different forms of Liostridia based on wall thinness and size. Liostridia ternata, this squash form, is kind of interesting, well, for other reasons. There are some Lophostridians, I put that in quote because it's more like, not like applied sculpture the way we think of in the Phanerozoic, but these are more sort of patterned wall structures. A lot of material we can dump into Nucelostridium. That was a Timofeyev genus. It's sort of convenient, but it groups together lots of things that have a larger outer wall and then sort of an inner blob associated with them. And then at the bottom, you see interesting things, these sort of archaeoellipsoides type stuff. Some people have thought that these might be aconites. I think that's unlikely, that they're cyanobacterial. And then various other shaped, test tube shaped things, and on the way to kind of base shaped microfossils, I suppose. Here's some filaments that we have. If that Proterocladus is real, then that would be evidence for crown group green algae, but most of these are sort of either problematic or they're simple spheres. So simple sheath material. So for instance, these microbial mats, question mark, these are intertwined bits of Siphonophycus, or in some cases, they look sort of like Eomycetopsis, but basically either twisted or straight sheaths. And then this Eohalotheke is also considered to be a cyanobacterium. And there's some other interesting things here. Valeria lophostriata, pretty well known throughout the Neoproterozoic anyway. Here it's occurring in fresh water, which probably indicates that Valeria is a whole group of organisms and not just one particular species, since it's been found associated with many marine sequences. And pretty good preservation. Looks like we have some things germinating, like in G here. So some interesting preservation is quite good for pathological. This is not from thin sections. These are all from maceration. Some potentially glenids, I'll skip by that. And then this is sort of interesting. Some of these forms look like they have perhaps chytrid damage, or in other words, they would be examples of early fungal activity. Now we move to what we really want to talk about, which are these clusters. There are different kinds of clusters, varying from just a lot of cells bunched together that you see in B there. Symplaslosporidium, I think another Timofeyev genus that we've seen mentioned earlier in Kath Gray's talks. These cell sheets, this is kind of interesting where you have flat sheets of cells. These look like they might be on their way to some kind of hybridictiacean form, but we don't have regular numbers and the way we do in the Phanerozoic. Various kinds of linear chains as well, and then these are all 10 micron bars. I should mention that, except for this big guy, these two elliptical things where it's 100 micron bar. So we find a whole range of interesting things here. And I want to focus on this morula form. These are cell clusters. originally I've referred to them as, well, sort of, I call them not Satka. They're not really Satka, but they're Satka-like. But they turn out to have some characteristic forms. They tend to be clusters of elliptical shaped cells. They're sort of kind of merged together, often not quite distinct in terms of the walls associated with them. So it makes you think these don't look like typical walls of, say algal clusters or individual cells that have just sort of fallen together. They can sometimes tend to have a relatively low number of cells, 16 to 20, centrally clustered, and they can occur with or without a diaphanous membrane. So here we have another. Hello?
You have 2 more minutes, okay?
I have what?
3 more minutes.
3 minutes, okay. So here's some examples of these shapes, sometimes with little associated membranes around them. And basically what we've done is we've compared these with another equivalent age example of something that was described in that June paper from the Diabeg Formation in Lower Diabeg. And this is what we think is a protist that shows cell differentiation and also a differential adhesion to create a multicellular type of structure, a multicellular structure that has 2 different kinds of cells in it. And this is the first example of this kind of organism and this sort of level of organization that's occurring at about 1 billion years ago. Here's what these look like in real. These are all in thin section form, and I don't have time to go over the details, but you can see on the left, these are simple clusters with different shaped cells. And then on the right, you see the more mature form that has an outer wall of elongate cells and an inner layer of isodiametric cells. It turns out that there's some deposits, in particular this one on Elan Flade this is all northwest Scotland, that have been extracted by palynological methods, and they are similar to this form in the left that comes from Lower Diabeg. So we think we have the palynological form as well to this form. We've put together a life cycle for this form, which involves multicellular, multinucleate forms that then cellularize and produce these cell balls, and then the cell balls undergo maturation and finally produce the cyst, which we see. Here's sort of the crux of the matter, though. The living form that we're comparing this to has a very complex life cycle, and it includes this unit 4, this sort of morula-like form that is seen here in living forms of this organism, Creolimax fragmentissima. There's also a similar related form called Spheromorpha artica that has a similar thing. So basically our conclusion is that this type of morula form is a form of a reproductive structure that's related to these holozoans. And just here are some examples of comparing the SEMs from the lab in Barcelona, the multi-genome lab, Inaki Rees-Trillo. And here's our conclusion, which is that we have these forms that are morphologically similar to the life cycles of ichthyosporine holozoans. And these, if that is true, then we might be able to say something about both about evolution of holozoans, but also something about the trophic relations in these early freshwater communities at 1 billion years ago. And the conclusion would be ultimately all is not algae. Thank you.
Okay, thank you very much, Paul. Very nice talk. Do you have questions for Paul?
There are no questions in the question boxes, unfortunately, at the moment.
Okay. So move on for the next standard talk. Now we're gonna listen to Yogmaya Shukla, and she will present the lecture Microfossils and Age Controversy of the Rocks of Kurnool Group of South India, a debate. So, Yogmaya.
Yes.
Hello.
Hello. Hello.
Hello, we can hear you and we can see your presentation.
Okay, that's very good. So very good evening to all of you. And today I'm going to
in the full screen mode.
Okay.
Okay, nice. Thank you very much.
Okay. So today I'm going to present my study on microfossils and age controversy on the rocks of Kannur group of South India, a debate. This study has been conducted by myself and my supervisor, Professor Mukul Sharma. And as you all have heard about the issue of age fossil inconsistency frequently occurred in the Precambrian sedimentary succession, and so is the case in Indian Precambrians also. And in the absence of reliable dates, and the data on dates are actually lacking, so the age of these basins are poorly constrained. And a very famous example of this age fossil inconsistency is Vindhyan Basin, where Cambrian age fossils have been recorded from Paleoproterozoic sediment. So this is a big issue which imposes some challenges in deciphering the age of the basin in the absence of direct radiometric dating. Here I will confine myself to Proterozoic basin, which lies in the southern part of India. There are two basins, the Kannur basins and the Bhima basin. They are supposed to be coeval, and they are supposed to be connected at the time at the Precambrian times, and they both have same lithologies and having alternate clastic and chemogenic sediments, and they are traditionally regarded as Neoproterozoic on the basis of paleobiological data and the age. So this is the Kannur basin. I will confine myself to this Kannur basin. It overlies, unconfirmably overlies on the sediments of Kadapa basin. This is a crescent-shaped Kadapa basin, and this is Kannur basin. There is an angular unconformity between them. The duration of the hiatus is not known, but there are several magnetic and tectonic events between 900 to 1100. So that's why this basin is traditionally regarded as Neoproterozoic. So this is the entire succession of Kannur base group, and it has been divided into 6 formation, the Bangan-Palle quartzite and Naji limestone and oak shale, and then second cycle of quartzite, limestone and shale formation. And the depositional environment is overall considered as shallow marine environment. And the first indirect date came from this Bangan-Palle quartzite unit. This quartzite unit is actually diamondiferous, and the source of diamond is considered as the nearby Vajrakud kimberlite. So in 1973, some workers have dated this kimberlite, and they said that the age of kimberlite is 1114 million years. So the entire succession should be younger than that, and they have considered this as a Neoproterozoic basin. Similarly, after that, Murthy and Raman, there are a couple of workers also studied the shales of the basin, and the age they have obtained lie between some 980 million years to 540 million years, and they also said that Kannur group is actually Neoproterozoic in age. After that, a study in 2008 and 2010, 2 papers have come, and they have said that the Kannur group is should be Mesoproterozoic, and it should be older than 1100 million years. The basis of that is they have dated limestone denolith, and the nearby sedimentary basin in the vicinity of that kimberlite is are the 2 basins, the Kannur basin and the Bhima basin. So Kannur, as you have, it is shown here, the Kannur has 2 limestone formation, Nazi limestone and Koel Kuntala limestone. So they have said that some part of from these two formations got entrapped into the kimberlite. So the entire succession should be older than 1100 million years. That is the date of that kimberlite, the Sindhan-Palli kimberlite. So the limestone xenolith has been considered to be the part of the sedimentary rocks, and on the basis of the age of the kimberlite, it suggested late Mesoproterozoic age, older than 1090 million years to the Kanul and the Miwams. Prior to our study, there are several paleobiological reports from the Kanul basin. The first report came from King, and he mentioned some circular structure, and he then at that time called them as the cycloid scale of fishes. Now they are considered as carbonaceous, very famous carbonaceous structure, Chuaria. Similarly, after that, Ajoker also recorded some Palmoria-like carbonaceous remains, which are now grouped into a formal taxon, Chuaria. After that, Vijayan and Rao, they have reported some bioturbation structures and worm burrow-like structures from the Naji limestone. So Naji limestone is also a part of Kanul subgroup, and it underlies the oak shale. After that, Saluja et al. Have recovered some omnivore microfossils like Leospiridia, Lophospiridium, and also some Ptenomarginata like Etritas. Subsequently, in 1999, Sharma and Shukla recorded some carbonaceous remains like Chuari, Tawed, Elopsophysic remains. And they also consider the age of the group as Neoproterozoic. In 2008, some Ediacaran elements like structures like Thuitia disc and Cyclomedusa species from the Penium Quartzite formation is also reported. So the paleobiological data suggested the age of the group is Neoproterozoic, whereas the indirect dating of some of the sediments of Kanul group said the age is Mesoproterozoic. So the controversy still remains there. And then we have performed a detailed paleobiological studies, and as this succession is deformed, mildly deformed and unmetamorphosed sedimentary basin, and fossil assemblage suggests Ediacaran age. So what should be the correct age of the Kanul group? So we have used paleobiological information and Proterozoic microfossil as a tool to decipher the age of Kanul group. And we have performed a detailed systematic study, and we have collected samples from oak shale of Kanul. This is the fossil bearing locality, and below the oak shale, there are some Ediacaran elements were reported previously, and from the above unit of oak shale, some Ediacaran elements are also have been also recorded. So we have performed and we have obtained moderately diverse assemblage consisting of 21 species belonging to nine genera, and this include some forms like Navifusa and Jacutinima and Siphonophycus. And Jacutinima, as we know, it is commonly known from Neoproterozoic successions. Here we have placed Naviform vesicles for tapering ends into Navifusa, and botuliforms cells without an X, with or within or without chain, are as Jacutinima. So this is also
have 3 more minutes. Okay.
Yeah.
You have 3 more minutes.
Okay. So these forms are actually typical of Ediacaran. Similarly, we have also obtained some more forms that belong to Neoproterozoic succession when we correlated globally. And some Acritarchs are also recorded. And as we all know, we have also recorded some elements of ECAP flora like idiacaran complex acanthoform phelanoflora like Variomargospiridium and Cava spina, and ELP assemblage like idiacaran phelanoflora. We have recorded 2 species of that, Leospiridia crassa, Leospiridia jacutica. So these are also known from meso to neoproterozoic succession. And we recorded as a very good assemblage of very distinct idiacaran element, that is Obrushuela. We have recorded 4 species of Obrushuela, and we have recorded a very good number of these specimens. And these are recorded from idiacaran to Cambrian silicified and ornate-walled assemblage from different localities of world. And the neoproterozoic idiacaran acme is the real phenomenon for this assemblage. So this is the distinct idiacaran element we have recorded from Kannur microfossil assemblage and in numbers of. So affinity, this assemblage is belong to non-sentient filamentous forms, some cocoidal forms, some colonial forms, acritarchs, and possible eukaryotes. We have recovered a decent assemblage of 21 taxa. And this is the list of microfossil taxa reported from Kannur basin. And the biostratigraphic significance of this assemblage is when we correlate it globally, there are 2 or 3 types of forms. Some are of typical mesoproterozoic to neoproterozoic type succession, and some are distinct or element of neoproterozoic or even idiacaran succession, like Variomargospiridium, Cava spina, Obrushuela, and some species of Obrushuela. So here we can draw a conclusion that in the essence of geochronological dates, we can use paleobiological information to decipher the age of the basin if they are not in conformity. And Kannur basin have distinct idiacaran elements and indicate the presence of some pre-Cambrian biotic element. So there is a need for elaborate exercise on generation of geochronological data or of Proterozoic basins of India, so we can solve this age fossil inconsistency. Thank you.
Okay, Yogimaya, thank you very much. Very interesting presentation. Do you have any question for Yogimaya?
No, there are no questions yet.
Okay. So thank you very much, Yogimaya. And let's go to the next presentation.
Evelyn,
yeah.
do you want me to introduce the person?
Okay, please.
Okay. Fairchild speaking, and I'd like to call Claudio Gauscher and his colleagues to present the paper on eutrophic conditions prior to the end cryogenian glaciation in the Otavi group, Namibia, a causal relationship.
Presentation में जो problem थी समझ में आ गई.
Hello.
Okay.
Can you see me?
Yep, we can see you and we can see your screen. Yeah.
Okay. I don't see my own screen. I don't know how. Let me just find where I can see my own screen. Sorry. So you can see my screen there?
Yeah, we see your screen. If you make your presentation, we can see it.
Okay. Now. Okay, thank you very much for the organizers to allow me to present this talk, and especially to Thomas Fairchild and Evelyn Sanchez, conveners of this Precambrian session. So I would like to present you some data from the Otavi group, the cryogenian of Namibia, and some features which allows us to infer eutrophic conditions just prior to the end cryogenian Gaub glaciation. So the Neoproterozoic glacial deposits were recurrent in the cryogenian and in the Ediacaran. So actually here we have 3 glacial events shown, but there are probably more. We are going to talk about the largest of all these, or the most severe of all these glacial events, which is the Marinoan. In Marinoan, it is called in Australia, but it's a term that has been used a lot. And it happened around 635 million years. And we are going to show you some data from here, from Southern Africa, from Namibia. Here is a geological map, and here in blue, you can see the Otavi group. And the Otavi group has a range of sedimentary environments, all marine, but ranging from a shelf here, more in the north and in the east, to slope here in the south and in the west. We are going to show two sections, one here at the Keilberg section and the other near Fransfontein. So already we published already quite a lot of information about these sections. These are the two papers, mainly about chemostratigraphy. So I refer to you to this paper for more for the. These are the stratigraphy, more the shelf section up to 1 kilometer in thickness. And here, this is the four slope section near Fransfontein. You can see it is less thickness, half more or less. Also interesting, we have here two glacial levels here at the base, the Chuos formation, which is around, which could be cryogenian or even Tonian in age, because the age constraints here at the base are 746, so it could still be around 740, but this is not the subject of this talk. The subject of this talk is the Gaub formation here, more or less in the middle of the Otavi group. And you can see both sections. This is the diamectites are well represented, and the rest of the section is more or less carbonates, so dolostones or limestones. And we sample all these sections. I am showing you here as dots only the fossiliferous samples. So I will just go through the stratigraphy and show you some outcrop photographs. Here, I will start here at the Gauss-Auros formation and going up to the Gaub and Meyerberg formation. So from the base to the top. First, the Auros formation at the near the base. You can see this fine-grained dolostones and also limestones. They are organic rich. Here, the corresponding unit in the fourth slope, which is the Ombatier formation. And you can see here there's rhythmites, limestone, dolostone rhythmites, sometimes also with quite a lot of organic matter, up to 2 or 2.5%. Near the top of these units, we can see a shallowing upward trend where the limestones get reworked, eroded, and redeposited as breccias, sometimes oolitic limestones like these clasts here, and leading up to the diamectites, glaciogenic diamectites, which are part of the Gaub formation. And you can see here lots of carbonate clasts, but also a few basement clasts. And you can see here, this is the Fransfontein section. You can see the drop stones, very nice. This has been published already, of course, by Paul Hoffman and co-workers, but we are working in the same sections. And on top of the Gaub formation, here you can still see the diamectites. We have the so-called cap carbonates, and these cap carbonates or cap dolostones are already the base of the Meyerberg formation. Here is where all these famous structures which giant ripples and all this stuff has been described. So these are the typical cap carbonates of the Neoproterozoic glaciations or glacial rocks. Higher up in the stratigraphy, we see that there is again a deepening upward trend, and this deepening upward trend we can see it by this finally bedded or finally laminated sometimes limestones, which are still part of the Meyerberg formation. So what about the fossils? So we can distinguish three assemblages here. We are going to talk more about these two, because most of the people that worked in these rocks have focused on what happened after the glaciations, and rarely someone has studied what happens before the glaciation to try to solve the mystery of the causes of these glacial events. So in fact, we have these three: this Bablinella faveolata assemblage, then which is below the diamectites. The diamectites show another assemblage, which is more, we named that Asteridium tornatum assemblage. And then on top, there is a different assemblage, which is characterized by Octaedrixium. So just to show you the Bablinella assemblage, Bablinella faveolata, also called Spherocongregus variabilis, it's probably a cyanobacterium, and it's made up of these colonies. You can see it here. This is with transmitted light, and this is with the reflected light, and you can see the microcells that made up the fossil here with some sort of folding. You can see it here with transmitted light. This is quite simple, but they can also be attached and form so larger groups of colonies like this one. Here again with transmitted and reflected light, and here chains of the same colonies. Please note that they are carbonized. This is anchimetamorphic conditions, and that's why we also have this reflectivity, which is quite important. We have also larger groups of colonies like this one, comprising maybe a few dozen colonies, maybe 100 in some cases. So this Bablinella assemblage is typical of the whole package of carbonates below the diamectites. Here, one example where we can see the more coherent colonies like this one, but also other colonies which are characterized by larger cells, and they are less tightly packed, so they are actually loose, and also some loose cells here. This has been described before for Spherocongregus by Mary Moorman already many years ago. So in thin sections, we can also see the Bablinella acritarchs, and here one example. Of course, here this is with reflected light. Again, here one fragment. So it can be very easily determined also in thin sections, because as you see, these are carbonates which are translucent. So it is easy to look at them in the thin sections. So in the Gaub formation, we have a different assemblage which is a little bit more diverse. Here, Asteridium tornatum, you can see this short and robust process here, which here in transmitted light and here in reflected light. Here the same. The other specimens here only in transmitted light. Always this very short process, but robust, and the small size is also typical, about 15-20 microns.
Claudio, you have 3 more minutes, okay?
Oh, only 3 minutes, okay. On top of this, we have Octohedrixium in the Mayeberg formation, but I'm not going to get into that. What we did a quantitative analysis by point counting on the thin sections and taking an area of 140 by 90 microns in each point and counting down to the whole depth of the thin section, how many Bablinella colonies are there. So we did more or less a census of all the statistical census of how many colonies per cubic centimeter we have. And then we measured in macerations the mean and maximum diameter of the colonies. Of course, in the Gauss formation, we did it in the matrix. Here you can see that there are fossils in the matrix, and you can distinguish them from the ones in the clasts. So what we found here, you can see the graphs. The concentration, the fossil concentration in millions per cubic centimeter goes down from the base to the top. So they are very abundant at the base of the Gauss formation and the Auros formation, but at the top they become more rare. And the diameter of the cells, of the colonies, sorry, does exactly the opposite. So it goes up. So from the maximum diameter goes from 15 or 17 microns up to 40, and the mean diameter more or less the same. So here you have the total organic carbon also. So how can we interpret this? So we have a high bioproductivity interval there, bioproductivity collapse, which is also concomitant to negative carbon isotope values for the carbonates, and then glacial conditions, and of course then a post-glacial stage which we are not going to talk about. So the trophic conditions are dominated by Bablinella. It's only one species, and very small and very abundant. So this we interpret as a high cell division rate and a high TOC and high carbon isotopes ratios. So the collapse is still very low diversity, monospecific, but now the Bablinella are very large and scarce. This is different to the previous stage. Carbon isotopes go to negative values and very low TOC. And in the glacial conditions, this dominance is no more, so we have more 3-4 species, and Bablinella becomes small again and scarce, and we have the chemostratigraphy shows more oxygenated conditions. So Bablinella is a morphologic and ecologic analog of microcystis. Here at the right, so photos of microcystis present day, and here Bablinella from the Otavi group. And microcystis is known to cause large blooms, both in lakes and at the coast. So we applied this model, and we consider that during the pre-glacial stage, we have high bioproductivity, blooming population of Bablinella, which generates a lot of organic carbon, which is exported to the sediments. This consumes CO2 and produces dimethyl sulfide here, which promotes the formation of clouds. So this is pre-glacial, and it tends to cool the climate. So this is probably what's leading up to the glaciation. During the collapse, then we have probably a CO2 limited bioproductivity. So we have very little biomass here, and advance already of the ice and the regression that we have seen in the photos. So, and then during the Gauss glaciation, full glacial conditions and some reinvigoration here of circulation, which makes for more oxic conditions, and the bioproductivity goes up a little bit. So what we think is that this situation could be promoted by fertilization of the ocean, especially by iron. We have in the Neoproterozoic banded iron formation deposition, and this here in the Otavi group, we have iron formations right at the base, so it is not it is probable that we had this iron dissolved and promoting the bioproductivity. So why is that? Probably because at that stage there was the most intense rodinia rifting events here right near the Kalahari Craton, and this might have pumped a lot of iron and other nutrients into the hydrosphere. So that would be all. Sorry for the last part. It was quite a little bit of a hurry, but I hope that it could be understood. Thank you.
Thank you very much, Claudio. Someday we'll have to talk about this together. The next and final talk in this series is by Uyan Zhou Xiao Iguan, and it's entitled Ediacaran Acanthomorphic Acritarchs in the lower Doshan-Tuo formation of the Yangtze Gorge area, South China. Distribution and stratigraphic implications. Thank you for your participation.
Can you hear me? Hi. Can you hear me?
Yes, we can hear you and we can see your screen if you just want to make your presentation into the full presentation mode.
So is
Perfect.
it correct now?
Yes, that looks great. Go ahead, please.
Okay. Thank you. I'm Qin Ouyang from Chinese Academy of Sciences. Thank you for this chance of presentation. After this very interesting record from cryogenia, I'd like to bring you back to Ediacaran to see how the post-glaciation aqueducts could contribute to biostratigraphy. Here's a chart showing what we've got for Ediacaran biostratigraphy. These are all globally distributed eukaryotic fossil groups of the Ediacaran period. You can see that for the upper part of Ediacaran, there are various fossil groups, but for the lower part, only acritarchs. So this is why Ediacaran biostratigraphy relies so much on acritarchs, and especially acanthomorphic acritarchs. Up to date, Ediacaran acanthomorphic acritarchs have been reported from 9 places around the world, and I hope this number will increase in the future. The first systematic biostratigraphic work was done by our keynote speaker, Dr. Gray, in Australia, where 5 biozones, including 4, based on acanthomorphic acritarchs, were proposed. And to date, this is still the only biozonal framework for the Precambrian strata. So now let's turn to South China, where the Ediacaran succession also yield abundant acanthomorphic acritarchs. The Ediacaran succession in South China is highly fossiliferous with good age constraints. The Doshan-Tuo formation that yield abundant acanthomorphic acritarchs was developed on a ringed carbonate shelf, as shown here. And microfossils that were preserved by silicification and phosphatization have been reported from the inner shelf and outer shelf areas. Here are the Doshan-Tuo rocks and chert nodules, where we get the silicified microfossils. Here are some of the published acanthomorphic acritarchs from the Doshan-Tuo formation. After studying them for almost half a century, now there are more than 100 species, and people began to find that for different stratigraphic intervals, there are different fossil composition. So this is fundamental to biostratigraphy. Actually, in recent years, there are several different biozonal schemes proposed for the Doshantuo formation. However, there are something very important that we haven't done in South China yet. So let's see how things were done in Australia. Fossil ranges for each drill core were first recorded separately, and only those species that occur at multiple drill cores were then selected for correlation. However, in South China, we usually compile data from different sections into a composite section. So these two steps in the red box were missing. So we conducted an investigation on the distribution of these acanthamorphs to see how many of them really have a global, a regional distribution, and are there occurrence or abundance data really correlatable among sections. So we selected 3 sections here, the blue stars, Jiulongwan, Jingguadun, and Wuchishan sections in the Yangtze Gorges area, where the Doshantuo formation has been intensively studied. So the little stratigraphic sequence of these 3 sections are identical and can be well correlated. In this area, the Doshantuo formation has been subdivided into 4 members. And our focus was member 2, which directly overlies the member 1 capitolostome. Here are the little stratigraphic sequence of the 3 studied sections. The sample strata at Jiulongwan and Jingguadun are roughly synchronous with each other, representing most part of member 2. And the sample interval at Wuchishan represents only the upper part of member 2. So we collected the church nodules and made them into thin sections. And here what we've got for acanthamorphic architects. Totally, we have 1,574 specimens, from which 24 genera and 69 species have been recognized. Here are some of the new forms we found from the new materials. Some are new species and some are named yet. And first, let's have a look at the spatial distribution of these acanthamorphs. We can see from this Venn diagram that out of the 69 species, there are 41 that occur at only 1 section, including the 4 shown in the last slide. So you can't apply these 41 species into correlation, because how? And fortunately, there are still 12 species that occur at all 3 sections. Maybe there are common elements of the member 2 acanthamorphs in this area. They include the Appendispero grandis, which has a global distribution, and Tianzhushania spinosa. This is a very well-known species among us who studied the Doshantuo materials, and these small spiny architects with relatively small vesicle and densely distributed processes. And when we put the published data in, we found that among the 12 species that occur at 3 sections in our study, 9 have been reported before. And also among the species that occur at 2 of the 3 study sections in our study, there are 7 species that have been reported from at least 3 other sections in previous studies. So now we have 16 species, that is 9 plus 7, that occur at more than 4 sections in this whole area. They are listed here, and we consider them as regionally distributed. So now we try to find a stratigraphically useful record for these 16 species. So we checked the first appearance data for these 16 species, and we found that some of them here in red, their first appearance are consistently low. Actually, lowest among all acanthamorphs in the Doshantuo formation, very close to the base of member 2, or say the top of capitolostome. And there's another group here in green. their first appearance are consistently higher than the first group. So the first appearance data for these two groups of archaeomorphs might have some potential in stratigraphical correlation. And we also checked the variation of relative abundance for each species at each section. And we found that this might have some stratigraphic useful record too. For the lowermost part of number 2, at only Zhoulongwan and Qingguadun, the Tianzhushenya species here, the gray bars, take the dominant role. And upsection only about 10 meters, although Tianzhushenya species still are dominant. This Lyciella brevis, its relative abundance increases significantly at what? Sorry?
You have 3 more minutes. Okay.
More minutes for your presentation.
Oh, okay, thank you. So for the up section, the relative abundance of Tianzhushenya species decreases, while these small spine acritas, their relative abundance increases. So this might be stratigraphically useful too if can be confirmed by more data from other sections. And we also found that there might be a rise in taxonomic evenness from lower member 2 to upper member 2. You can see that from this Pylos evenness index, the upper 3 all have higher values than the lower 2. And all that I talked about was how this data could be used to regional stratigraphy. And how about global correlation? Well, one unfortunate fact is that there are not many species that occur globally. And some of them, including Appendisphaera grandis and Cavaspina acuminata, their range are too long to be very useful. And how to interpret this? Well, the first thing might be the number 2 of the dose transformation might be stratigraphically older than other acanthamorph-bearing strata elsewhere, like in Australia or Siberia. But this can't be the only interpretation, because we know that taxonomy can be strongly influenced by taphonomic bias, and maybe there are some globally distributed taxa that we fail to recognize in some assemblages. And the third problem is that maybe there are some globally distributed taxa, but their abundance vary from place to place. So in some places, maybe their abundance are too low to be recognized from enough sections or drill cores. And also some species, maybe they are just geographically restricted. So we just need to know who they are and avoid using them. So here's the brief summary. What we now know is that some species are just more useful than others due to their distribution, and so it's not always a good idea to use them without a good investigation on a regional scale. And what to do next? Well, need more data, of course, and a taxonomical treatment that can reduce taphonomic bias. And also we need to focus on some like change in the assemblage composition rather than occurrence data of specific species. So this is all. Thank you.
Much. That was a very, very nice presentation. I believe that now we are coming to the end of our session. And we would like to thank all of the participants for their very, very interesting.
I think we've lost you there, Thomas.
Very informative talks. So thank you for your participation.
Okay, everybody, thank you for your participation. I think that's it what Professor Tom was supposed to talk. And thank you for your attention, participation. All the best for you. Okay, so that's it.
Okay. Well, I'll just say thank you very much to Evelyn and Thomas for making sure we had this Precambrian session. I found it fantastic. I love the Precambrian. I've only ever done one mini tiny project on the Ediacaran and found one acritarch. So I'm very jealous because I think the older stuff's really interesting, but I'll stick with the Mesozoic for now. So thanks again. Right, we will have a short break. We will be back in just under 15 minutes. So if we can be ready to sort of go in just under 15 minutes, that'll be great. Thank you again, Evelyn and Thomas. Thank you.
Okay, bye. Thank you. Bye-bye.